All right, guys. So, oh, where did it go? Interesting. Okay. I'm having some now technical difficulties of my own because I was sharing and then I escaped and then now I got to
You're
get
just trying to make me feel good.
get back to it. There we go. Okay. But now it's not. Okay, so let's escape. There we go. All right, gotta grease those wheels again. Here we are. So, again, we're going to be talking about eukaryotic microorganisms this week. So, today's lecture is just going to give you an overview of the different types. And then on Wednesday, we'll home in on the fungi more. And on Friday, we'll talk about the helminths or the parasitic worms primarily. This chapter is going to be looking at, first of all, what makes eukaryotic cells different from prokaryotic cells. And of course, we all know the differences because it's been um, discussed in several lectures. It's been on quizzes, on the midterm. So by now, you know that prokaryotic cells are different because they don't have membrane bound organelles, they don't have nuclei. And so the microbes we're talking about now do have those things. So a little bit of the chapter is going to be going into those structures. This is not cell biology, so we'll really just kind of gloss over it, but you do need to know these structures so that you understand the difference between prokaryotic and eukaryotic cells. We'll talk a little bit about fungus today. They're very important in terms of cycling nutrients and making that available to other organisms. They are decomposers, so they break down dead organic material so that it doesn't accumulate. And they also happen to be opportunistic pathogens. So they provide us all kinds of services like the decomposition, the nutrient cycling, the antibiotics, um, making of food and beverages like kombucha, but they can also make us sick as well, which we'll talk about more on Wednesday. We'll talk about algae a little bit. They're not very pathogenic. Some of them can be. Some of them can excrete toxins that can then be, detrimental to life, particularly in algal blooms, um, and then can also really suck up the oxygen and kill off fish and other aquatic life. What you're seeing here are some diatoms, which are these very, I think, beautiful um, living organisms that have these highly geometric shapes and structures. We'll talk about the protozoans like Giardia, which is, if you are into backpacking and camping and any kind of situation where you need to pump your own water, then you realize that Giardia is something that you could potentially get, which is why you want to make sure you have a good working pump or filter for your water. And then we'll get into the worms. Now, parasitic worms are tricky to treat, as are some of these other organisms, because if you guys remember when we talked about microbial control a few weeks ago, prokaryotic cells have different mechanisms in their cell. And if we target those things, it's selective. Now we're talking about pathogens that have the same types of cells as we do. So if you have a parasite that has very similar biology to the host, then when you try to interrogate those biological processes in that parasite, you can have downstream effects of also interrogating the host's biological processes. Okay, so you guys should remember that bacteria have been around for 3.5 billion years. The eukaryotic organisms arose about 2 billion years ago. So the archaea and the bacteria had free range for quite a while. And then all of a sudden, we start seeing a symbiotic relationship where two prokaryotic organisms come together. Okay. And that's how a eukaryotic cell arises. It now has organelles that were once an actual prokaryotic organism that was engulfed, internalized into another prokaryotic cell and co-opted and used. Okay, and one of the early and most easy to grasp examples would be chloroplasts. Okay, so chloroplasts, as you guys know, are organelles that are in plants and cyanobacteria that allow photosynthesis to occur. So it's an organelle in that prokaryotic cell that now turns it into a eukaryotic cell and allows it to get energy, right? And what's the payoff? Well, now the chloroplast has a safe environment inside this other cell. And so this is kind of the demonstration of how that works. We have a large prokaryotic cell, and in that cell, maybe this is an archaea cell, maybe it's a bacterial cell. But what happens is a smaller prokaryotic cell comes into contact and it starts engulfing that smaller prokaryotic cell. And for whatever reason, that cell isn't destroyed inside, whether it's protected by a vacuole or it has defenses against the digestive enzymes in that larger prokaryotic cell. It is now safe inside this wonderful environment, protecting it, blanketing it from the harsh outside world. And it has everything it needs. It has nutrients from the back from this other larger cell. It has the metabolic pathway components to start dividing, right? And so now that single cell turns into multiple cells. And this is thought to be how some of these prokaryotic precursors came to get things like mitochondria, which are the powerhouse of the eukaryotic cell. Okay. These small prokaryotic cells evolve over time into other structures like the Golgi, right? The endoplasmic reticulum. And we'll talk about those in more detail soon. Until finally you get down this evolutionary tract into these protozoa or these algae organisms, okay, that then go on to give rise to things like plants and other more complex life. Okay, and so that is how it's believed that eukaryotic organisms came into existence by two prokaryotic cells getting together, giving it a whole lot of time to change, and then resulting in a cell that now has these different stations for different functions. Okay, so this is the eukaryotic cell. This is a very typical diagram, and it differs from prokaryotic cells in a lot of ways that we'll talk about. But remember, this came from a prokaryotic cell. And as I've said before, Mother Nature does not like to reinvent the wheel, right? So it's going to just keep what works and modify things that allow it to be more successful. And so you will still see things like glycocalyx, right? The slime layer, or what forms the capsule on the outside of the cell. Still has a glycocalyx in some cases. What's different is that now we're talking about the presence of nuclei, right? So that in prokaryotic cells, you just have nucleic acids bunched together in chromosomes. But now we have this nucleus here, and that has all the DNA. It has a nucleolus, which is a site for ribosome synthesis, right? Because the ribosomes need to be created somehow to make proteins from the RNA that comes from the DNA, right? The RNA is going to be in that nucleus, and it's going to then translocate out of the nucleus via the pores. And then, of course, like prokaryotes, it also has chromosomes, right? The DNA coiled up into different sections. Okay. So similarities and differences between prokaryotes and eukaryotes. Okay. Now let's get into the meat of really what makes them different, the organelles. Okay. So with the organelles, we have things like ER. The ER is going to be this stuff right outside the nucleus. Okay. That ER is going to be made up of rough ER, which is generally closer to the nucleus, and then smooth ER further out. Okay. The rough ER is going to take that RNA, and that's going to be the site of protein synthesis. So the ribosomes are going to translate the RNA into protein. And then in that rough ER, it's going to fold the proteins into their conformational shape that confers their structure. And then it's going to, tag them and label them, put, vesicles around them to shuttle them to the next organelle, which is the Golgi. Okay. And the Golgi is where that's kind of like the mail station. The post office, I guess, is what they call it, not the mail station, the post office, where the Golgi takes those proteins and further labels them, or if they're misfolded, recycles them to their respective areas. Okay, so it says, so think of a protein as an incoming package. It looks at the package, it says, okay, this is what you're designed for, and it shuttles it to the proper location, whether it's going to be a cytoplasmic protein or a transmembrane protein, that kind of a thing. The smooth ER is used, so the smooth ER is right here. That stuff is going to be used for synthesis of fatty acids and phospholipids, important things for overall cell homeostasis. The mitochondria, where's the mitochondria? Here's the mitochondria. The mitochondria are where ATP is derived and is used for energy to help the cell do its functions. And then in some cases, but of course not all, there are going to be these chloroplasts that have these stacked thicolloids, and that helps, not help, it does photosynthesis. So not all eukaryotes are going to have chloroplasts, right? Just the plants and the algae. So that may or may not be there in a typical cell. And then in some cases, there will be flagella. Right, where's my, let's go back to the draw. Here we go. So a flagella right here, or cilia, to help it move around, especially if it's a single cell eukaryotic organism. It's going to need to move. But not all eukaryotic single cell organisms can move. And we'll talk about that later on. Okay. Oops. Finally, we have the cytoskeleton. Okay, so the cytoskeleton is what gives it its shape. The cytoskeleton is made up of things like microtubules and microfilaments, and these things will polymerize and elongate to help the cell move for motility. And that will use things like actin and form pseudopods. It will also be the train tracks for translocation of proteins to their destination. So how does a protein get from the rough ER to the Golgi, and then from the Golgi to the area it needs to in the cytoplasm or to the membrane? It hitches a ride on the microtubules. Okay, it's not just floating around randomly and hoping to get where it needs to. There's precision. Any questions on this, guys? Again, it's not cell biology, so I'm not really going to focus on, well, what's the function of the mitochondria and the Golgi and the ER? Okay. But there might be a question on the final where you need to know what these structures are. Okay. Or you'll need to label these structures. So be familiar with this cartoon or a cartoon like this and what these structures look like. Okay. All right. Clear the drawings and let's go on. Okay, so the eukaryotic microorganisms that we'll be talking about: fungus, algae, protozoa, and worms. And then within protozoa, of course, there are going to be things like Giardia that I mentioned, and Plasmodium, that is the malarial parasite. So all of these things are different in their biology considerably. And we'll talk about that just kind of, in a rudimentary way, because, honestly, this could be a class all of itself. So it's kind of a glossing over of all of these things, but we'll do our best to cover it in a way that is informative. Let's start with fungi. Majority of fungi are unicellular, so think yeast. However, not all. They can elongate, and interestingly enough, depending on the conditions, some fungi can either be single cellular or multicellular. There are over 100,000 species that we know of, and we break down those species into basically two groups. There's the macroscopic fungi, the stuff you can see, think mushrooms, puffballs, the gill fungi that you encounter out on a walk. And then the microscopic fungi, the stuff we can't quite see. Now, that said, some of these in the form of mold, you can see when it's a composite, right? But if you break down that mold into the initial components, they become microscopic at that point. And the microscopic fungi are going to have two morphologies: hyphae, which we talked about when we did the fungus lab. So long filaments, long finger-like projections that we see with mold. And then yeast. The yeast is going to be circular or oval shaped. And most yeast is going to reproduce asexually. It's just going to bud off from itself. However, fungi, I've only really come to appreciate fungi recently. I used to think that it was my least favorite microbial topic because I was like, these guys don't even need to be pathogenic. They just do it, but they don't have to. So they kind of suck. But then I did this. I wrote this lab over the summer, and realized that, yeah, that is not a cool thing to do in the biological world, but fungus is so interesting in how it can be asexual or sexual. It can be hyphae or yeast, depending on the temperature, for instance. So that's called dimorphic. Okay, so if it's for instance a yeast, when it's in higher temperatures like body temperature, but then if it's outside the body at more of a room temperature, it's a mold or a hyphae. That's dimorphic. Okay, and so this is how these things become pathogenic. You have a mold that exists in the environment, and you breathe it in. And this was that this was in that clinical application section of the lab. You breathe it in, and then now it exists at a higher temperature as a single cell yeast. All fungi are heterotrophic, meaning what? If you're heterotrophic, what do? Anybody? It means you're not autotrophic, right? You don't get your source of carbon and things from yourself, photosynthesis. It means you need to be a saprobe, right? You need to be saprophytic. You need to secrete enzymes to digest things around you into smaller components that you then take and absorb in and utilize. Some, as I said, can be parasitic. They can live outside the body, like foot fungus, jock itch, that kind of thing. Some can live inside the body, like Candida albicans, right? When we think of yeast infections and things like that. And depending on the type of fungus, it's oftentimes going to dictate where it thrives as a pathogen. And because their distribution is so widespread, meaning they're all over the country, not over the country, they're all over the world. I mean, that means that they provide beneficial services everywhere, but they also can potentially infect us. And definitely when you go into warmer, wetter, more tropical areas, that's going to give these fungi more of an environment that they like to thrive. And then that's when you can get things like they talk about boot rot. When soldiers go to these tropical areas and they don't take their boots off, they get these really bad athlete's foot. Looking at microscopic fungi, what we have here is this colorful array of different fungus. And if you look at that through a microscope, what you'll see are these long filamentous structures, okay, that make up what's called mycelia or the mycelium. Okay, so all of these different finger-like projections or threads are the mycelia. And when you zoom out, what that looks like then is going to be this fluffy, cottony, cobwebby structures in the mold from a macroscopic view. Some mycelia will be will have a septum or will be septate, meaning there's a little division in kind of like bamboo, right? It has these walls in between, kind of sectioning off the mycelia into compartments. Other mycelia won't have this; they'll be non-septate, and so it doesn't have those delineations. This is looking at yeast, so it's going to be not the mold, not the multicellular. It's going to be a single cell fungi. That was a juicy one, Theo. Usually asexual reproduction. So what we have is the yeast cell here, and you can see in that yeast cell, look, it has all of the things. It's a simple yeast cell, right? A little tiny guy. But within that cell, you have a nucleus, you have mitochondria, you have ER, you have the Golgi, right? It's all there. And it will bud off from that initial mother cell into a daughter cell. And it will have all of the components that it needs to then grow and form its own unique yeast cell. Okay. And so it has to do that by, of course, budding at the site of the nucleus. So it has to contain that DNA. So if you can see this image here, it's budding, part of the nucleus buds with it, and then it leaves behind this bud scar, kind of looks like a potato in some ways, right? The eyes of the potato. And if this happens and there's a complete budding, then it stays as a single-celled organism. But sometimes the bud won't completely release itself. Okay. It won't completely release itself. And so then you get this long chain of single-celled organisms not completely releasing. And so we call that a pseudo-hypha or pseudo-hyphae, right? It looks like the hyphae, the filamentous hyphae of mold that make up the mycelia, but it's actually these single-celled organisms that haven't completely released from each other. Okay. This is looking at the two different types of hyphae. So vegetative hyphae is what's going to be used to digest and absorb nutrients. Reproductive hyphae is what's going to produce spores. Okay. And if you remember the fungi lab, there was that diagram that we went over during lab, and it talked about the life cycle and how it could be a single-celled, kind of like a yeast cell, and then that could asexually reproduce, or that could come together with the yeast cell of another individual, all right, so not clones, and then those could come together to form the gametangia, right? The gametangium, if it's just one. And then that forms into the hyphae, okay? And so depending on the type, it will have different functions. And the hyphae, the vegetative hyphae, is also what in some fungus will have a symbiotic relationship with plants, with the roots. So, oh, okay. So okay, we'll put your headphones on. So the mycorrhizae is an example of that. These vegetative hyphae provide nutrients to the root system of the plants, and as a quid pro quo, the plant shuttles some of the sugars that it makes via photosynthesis down into the roots of the plant and then into the fungus. Okay, so they provide benefits to each other. And again, with the yeast, it can be a pseudo hyphae if they don't completely bud off from each other. And then what we're looking at here below is septate hyphae. So this is a septum here. It's got these individual compartments with nuclei. Non-septate is going to be, still has the nuclei here, but there's no divisions in between. It's just one long like syncytium. Can you put your headphones on, please? Okay. Just a second, guys. Okay. No, put your headphones on. And here's another diagram of that showing basically the same thing. I think this is from the textbook though. So it shows you in the non-septate hyphae, you still have the nuclei, but they're not separated by this septum. Okay. So when we identify different fungal species, we can do that by using selective media, right? Different plates. We can do that by observing macroscopically for color and texture. We can look at it microscopically, right? By seeing if it's got the presence of hyphae or if it's single-celled like a yeast. We can look at things like physiological characteristics and whether or not they're pathogenic. And if they are pathogenic, where do they live? Where do they infect? And okay, well, then find something else. And then their genetic makeup, right? So we can do a phylogenetic comparison by looking at the sequence, the DNA. What? I need it out. Okay. Just 1 second, guys. I'll be right back. Sorry. Come here. Okay. So again, many different roles of fungi that is non-pathogenic, but we'll talk about the pathogenic roles again on Wednesday. They can be detrimental to our agriculture, right? If they are contaminating and rotting our crops or infecting our livestock, or being growing on our stored food, right? So if you have hay that you have cut and you let it dry out for a while, but then it rains on it or something, and you go to bale it up, that water that's trapped in there can lead to a bunch of moldy hay, and you don't want to give moldy hay to your animals, right? Or if you have a bunch of grain or corn that you store in a silo and it gets contaminated, then you're out that food. But while it has a lot of negative aspects, think of all the good things, right? Antibiotics, the production of alcohol, not just the alcohol that is consumed, but alcohol that is used for things like germicides, right? Different productions of acids like acetic acid, vinegar, different vitamins that we use and take as supplements. They break down dead plants and animals so that doesn't accumulate and lead to um, widespread infection, right? If you have a bunch of dead carcasses around, then the bacteria that like those kinds of things will grow in that. And then that makes us more susceptible to then contracting that because it's just in our proximity. Whereas if these things break down more readily and more quickly, there's less of exposure to that. And then making foods like kombucha, different cheeses, these fermented things, sauerkraut, what have you. And then also genetic studies. So yeast is a model organism in the laboratory that's used for all kinds of different genetic studies because it's really easy to use, right? It doesn't take up much space in a lab, and it's a eukaryotic organism. So you can do things in that organism and then translate that to human biology. Okay. All right. Any questions on that? That's the fungi. Any questions on that, on fungus? Any questions among us on fungus? No? Okay. Quiet group today. Must be just easing back into this. So now we're going to shift gears and talk about what are classified as protists. So protists are going to be the algae and the protozoa. Algae, unicellular. They can be in colonies, right? So when you see a bunch of them together, it looks like a film on the surface of water, but really that is not just one big plant, right? Because they're not plants at all. It's a aggregate of a bunch of different single cells, and they, like plants, are capable of photosynthesis by using chlorophyll, and they actually produce more oil than any other life form on the planet. And that's why a lot of biotech companies are using algae as an alternative source of fuel. The protozoa, also unicellular, and so therefore they don't have things like tissue, right? Because tissue is made up of a bunch of cells together. But even though they don't have tissue, they're eukaryotic, right? So their makeup in their cell, the nutrients that they need, the life cycle that they have is going to have similarities with other more complex eukaryotic organisms. Okay, so the algae have a cell wall. They may or may not have flagella. Okay, so diatoms are an example of this algae, and they do not have flagella, but other algal organisms may have a flagella, so it will be motile in the water. And again, like yeast, look at the inside and you'll see that they have the similar components that we talked about in eukaryotic organisms like us. They have mitochondria, they have a nucleus, their nucleus has a nucleolus, they have a Golgi, right? Now, difference, they have chloroplasts, which we do not, of course. They have other things like starch vacuoles that are slightly different, but for the most part, they are very similar to all life that is eukaryotic. Most of them, as I said, are not pathogenic, right? Most of them are free living, and they're going to be typically in fresh water, the kind of stuff that we see here, this green algae, but can also be in marine water and is a source of food for many organisms. So it is at the at the bottom of that food pyramid. Okay, and so other things rely on that, and then the things that rely on them indirectly rely on algae as well. They also, because they are photosynthetic, produce a lot of the oxygen that we utilize to survive. Okay, it's not just plants. Algae is also a great source of oxygen. Now, when algae goes wrong is when you have these red tides, which you guys have probably heard of, that they happen along the coasts in certain regions. And primarily these are going to be dinoflagellates. Some dinoflagellates are bioluminescent, so we can also see these beautiful images of at night on the coast with just this blue light along the shore. But other ones can secrete toxins that then harm the other life in that area. And then you get these large dead zones where nothing can exist but the algae. Okay, let's pick up protozoa on Wednesday. Any questions on fungus or algae? No?
Does colonial mean like it can be seen macroscopically, or just that
No.
it's together?
Yeah, colonial just means that they form colonies. So it just means that they, rather than them just being single cells, distributed throughout the ocean, they tend to coalesce together and form what looks almost like a large film. So it's colonial by that connotation. Okay, let's take a 10-minute break, and then at 11 o'clock we'll start with the proposal presentations. Okay, and so a couple of you have emailed me saying that you would like to go first, sooner than later, to get it over with. And so we'll meet up, and whoever wants to go, share your screen, jump into it. 5-10 minute presentation, very casual, no big deal. Don't stress out. It's just I'm giving you the chance to talk, and I'm going to be quiet. So let's take a break, and I'll see you guys at 11.